Thank you. Yes, I'm Mikey Ross. I head up the culture collection of Algae and Protozoa, and I'm a lecturer here at SAMS and Algal Biotechnology. So myself, Matt and Alice, we're all based at SAMS, and I'll give a quick introduction to who we are. Okay, so yeah, today the majority of what I'm going to talk about is about CCAP, which we've already heard a little bit about from Steve and Alan. Like I said, Matt, Alice, and I are from the Scottish Association of Marine Science. We are an independent marine research institute in Scotland. We are based in Oban on the west coast. We do a variety of different research here at SAMS, ranging from broad-scale physical oceanography and biogeochemical cycling, right through to biological sciences, algal research and stuff, which we're going to focus on. SAMS is a partner of the University of Highlands and Islands, which means that we can offer degree programs, undergraduate right through to postgraduate. We've got about 160 students all told. We also have a commercial entity, SAMS Enterprise, who basically leverage in-house knowledge for delivery of goods and services. Most pertinent to this call is about the seaweed farm, a seaweed nursery, and the Seaweed Academy, which is basically a kind of educational program. Most importantly for me, though, however, is that SAMS is also the host institute of CCAP, the Culture Collection. Okay, so CCAP, we are what's known as a biological resource centre. It's not a term that I'm particularly fond of, but essentially a good way to think of it is we're halfway between a botanical gardens and the Millennium Seed Bank. We receive funding from NERC, UKRI, as a large research infrastructure, a national capability service. So as Dan said, we're here to help. We're here to help properly. We're here to basically facilitate research, innovation, and enterprise the world over. We are essentially a repository for living strains of algae. We have 3,200 strains of microalgae, seaweeds, cyanobacteria, algal pathogens, and protozoa. So we're a massive kind of treasure chest of these biological resources. Our collection it's humongous, but it's also spatially diverse. This is a kind of map of where all the strains in our collection were originally isolated from. So we can see we've got representation from pole to pole and everything in between. So marine microalgae, terrestrial, freshwater, brackish, you name it, we've got algae from there. This pie chart shows a kind of breakdown of the kind of taxonomic coverage, so lots of chlorophytes and rhodophytes, diatoms, but also protozoa critically, which gives us our diversity. Steve also mentioned the kind of taxonomic tree of life and how diverse and polyphyletic algae are. The little CCP symbols there are where CCP has got representation across this, so we're a massive biological resource, incredibly biodiverse. This graph shows basically the growth of CCAP through time. So we're a living collection, we're a growing collection. Our oldest strain was first cited in 1892. It's Chlorella vulgaris, which Steve had in a couple of his slides. We've kept it alive ever since then. But we get new deposits each year from researchers across the globe, from our own team doing field sampling campaigns or inheriting collections who are folding for whatever reason. So what this shows us from the previous slides is that we are hugely spatially diverse, but also time resolved. We are basically kind of unique in what we do. So in terms of our facility, we are one of the oldest, one of the largest, and certainly the most biodiverse collections in the world. And what I'm going to show here is that we are having impact on what we do. These are colleagues for papers that have been published in Nature, utilising CCP resources. So we are having real-world impact in terms of both academic and commercial research. So what do we do with this collection? Well, basically we are a global point of access to this. So we are best known for sharing these cultures across the globe for supporting research, education, and commercialization. We also provide our kind of in-house knowledge for taxonomic services, so whether it be molecular or just morphological. We've got a cryopreservation facility, which means we can long-term safeguard these resources, avoiding things like genetic drifts or supporting biobanking efforts. With the cryopreservation facility, that also helps us to be a patent and confidential deposition service. We're regarded as an international depository authority, one of 3 in Europe, which accepts algal and protozoa material. So basically, we've got private collections where we can act almost as an insurance policy for your research. We offer consultancy. We have bespoke education programs, which we deliver both in person and online each year. And all of our goods and services are ISO accredited under 9001. We also engage with collaborative research projects, much like the Natural Products BioHUB. And some of our newest services include the Bioinformatics Gateway and Aries, which we'll talk about next. However, this means that CCAP, we've got such a diverse collection. We support a lot of different things. We've got an enormous remit from conservation and restoration, doing the good for the planet, for supporting fundamental and applied research, for supporting education outreach, supporting aquaculture. Ecosystem services, there's been a lot of talk about bioremediation already, right through to this kind of shifting sands of biotechnology, which Steve was talking about. We've got a huge remit and we are here to support this. So our involvement with the BioHUB, we are perfectly poised to be supporting not only the kind of agri-tech sector, but biotech generally. Now, we don't just collect stamps at CCAP. We want to know our collection like the back of our hand. We want to know as much about these algal resources as possible so we can offer the best services to you. So in the early 2000s, we began a systematic program of understanding the collection through DNA barcoding. So this initially was targeting the most important strains which we consider in our collection, so that oldest one, right through to ones which are supporting live feeds for agriculture. Through time, we've become more and more involved with genomic research. Our first whole genome was published on Phyratil, a diatom in 2008, and we're becoming more and more involved with genomic research projects such as the 10KP, the Darwin Tree of Life, and more recently we were involved with a collaborative project which increased the number of seaweed genomes available from 13 to 124. So it's really, really paramount importance that what we're doing. And we've also launched the gateway, which we'll talk about next. But as of today, we have about 96% of the collection which has got DNA barcode sequences attached to them. This is quite impressive given the lack of knowledge that we've got about a lot of these resources. But I envisage these final few percent is going to be the hardest. You know, they're going to be the most troublesome to get methods for and to get good sequence information from. Okay, the bioinformatics gateway. This is unique for a culture collection. It's basically a single point of access, free point of access to basically all of CCAP's digital sequence information. So what this platform does is it basically scrapes the internet for sequence databases, whether it be protein or DNA, pulls together all of the related information about CCAP strains to a single point of access for them. It takes all of that sequence information, which you can download, all the associated metadata with it as well. So it's a really, really powerful resource. Another new element of CCAP is ARES, the Algal Research Innovation Environmental Science Centre. This is something that Matt and I and other members of CCAP put together. Now, this was kind of brought about from a greater demand from our users for larger volumes of algae, different preparations of algae, whether it be slurries or freeze-dried, paste or whatever. More and more molecular services, more and more genomic work, and more metabolomic work, which Matt will talk about in a minute. So this was a kind of three-pronged centre. Dan used the words one-stop shop earlier. This is what CCAP is as well. It's basically CCAP goes large. We can offer a whole variety of different services to fulfil your needs. We are not unique in having scale-up facilities. Alice spoke about this already for CSAR in Swansea. However, what we are unique is having CCAP underpinning this and the willingness to kind of push frontiers and do different stuff. So with our facilities, we've tried out your kind of classic green and red microalgae. We've done diatoms, polar diatoms during the summer. We've done toxic dinoflagellates, cyanobacteria, unicellular, multicellular. We've even done seaweeds. We've done a whole lot of different things. And we've also got an analytical platform, which Matt and Alice will talk about next. Just an example of some of the ways in which CCAP ARES has been used. We've got some exemplar projects here. So on the far left is one that Matt and I were involved with Living Inc., who are looking into exploring alternatives to carbon black for algal pigments for textiles. Living Inc. are a facility in the States, which have got partnerships with Nike, Adidas, Regatta, Patagonia, so quite big players. The second on the left is a project which was with Ulva, so green seaweed that you get commonly, looking to strip out nutrients from wastewater and then apply that biomass to land. So Steve was talking about biofertilizer projects earlier, so this is comparable to that. The third one is a top masters project which Matt developed, and within that we had a whole module developed or dedicated to algal scale-up and downstream processing. So we're supporting education as well. And on the right-hand side, from one of our training courses a couple of years ago, Luna on the right-hand side there contacted us to say, hey, Mikey, can we try out our research in your facility? And we went, yes. And so we got Luna over here for 2 secondments to basically test out different growth media formulations and different lighting with the reactor systems. So the way I envisage CCAP ARES is happening is to be as accessible as possible for collaborative research, contract research, no matter how big or small, we're kind of open to it. However, I think that the best kind of case study for CCAP ARES usage is from one of my former PhD students, Ellie, who also coincidentally worked at Swansea for a while. She was looking into this cascading biorefinery for chromochlorus. Chromochlorus is viewed as an alternative to Haematococcus, which is in that world's natural richest source of astaxanthin, which is used in fish feeds and aquaculture. However, Haematococcus is quite challenging at times. It's prone to disease. It's quite a complex life cycle. Chromochlorus, while it doesn't produce anywhere near as much as astaxanthin, it's a lot more robust, grows a lot more quickly, and produces a lot more biomass. So what we done was to do a two-phase cultivation, phototrophically and then mixotrophically, to induce the stress response to basically pile on these carotenoids. And what Ellie done was to then use the analytical platform to do proximate analysis of the biomass through time. And you can see this shift from being protein rich right through to lipid rich. And the same was observed with the chlorophylls and carotenoids there as well. So it's a really nice way in which CCAP areas has been utilised. Finally, Steve mentioned a project that we worked on together about 15 years ago when we first met one another. This was screening the CCAP collection for lipid-rich algae. So in this kind of second-degree screen, there are 120 strains which were kind of characterized for the proximate analysis. What our colleague Fred has done is basically compile this amazing kind of taxonomic knowledge, molecular knowledge, with that compositional analysis to develop this heat map that we can see here. Now, this is, I would say that Steve's work was kind of seminal for what CCAP do. We get requests all the time for, hey, Mikey, which strain is rich in protein, which strain is rich in EPA, which strain is rich in whatever. And we always refer to Steve's paper for this. However, imagine we could do this for the entire collection, that 3,200 strains. How much of a game changer would that be? That would be an absolute game changer, not only for Algal Biotech, but the whole of algal science. And that's where the Natural Products BioHUB comes in. This is what's provided us the funding to enable this to be a reality. It's been my dream for this to happen, so I'm really, really delighted for that. I'm not going to talk anymore about that. That's more Matt analysis area, so I'm going to hand over, and thank you for your time.